Hello, welcome everyone to the UKME class for Molecular Genetics WebEx, where we're going to review the TissueEye Histopathology Online Tumour Assessment Module, Pilot Run 3, the lung cancer cases. We have two of our expert panel members here who are going to review the cases with you, and we have Dr. Perry Maxwell from Belfast and Dr. Jane Moorhead from King's College Hospital in London. So I'm now going to pass it over to Perry and Jane, and they're going to talk through the lung cancer cases.
Hi, thanks, Sandy. Okay, Jane, shall we start with the benchmark case? So the benchmark case was the case where we're presenting an previously annotated slide where we're inviting the participants to score the percentage tumour and the cellularity. So if we go to the annotation, just to show the annotations. Okay, we're not being able to show the annotations there, but the annotations that were previously marked up. Here we go. There we go. So these are the previous annotations marked. Let me just see if we can get rid of this. No. So you can see there the annotation was predominantly the whole tissue, and within that we scored the experts of 11 to 20% tumour content. If we go to a higher magnification and move around, we can see the scattered tumour islands with a background stroma and occasional inflammatory cells. However, the tumour is well dispersed, and that is why I think the experts agreed that it was 11 to 20%. Jane, any comments on that?
No, I'd agree with that totally, and also the reason it's marked up is because the tumour is so well dispersed throughout, so there's no area to enrich really. And also cutting the ends off looks like it's removing the bulk of the inflammatory response.
Yes, as we move across, you can see that the tumour is well dispersed throughout, and however, it is a minor population of tumour. There's no real concentration of tumour where we could highlight and enrich to get it greater than 11 to 20%. So if we move on to the cellularity, the cellularity really gives us a means of determining the starting DNA for any methodology, downstream methodology, and the experts estimated the cellularity as low. This is typical of a small biopsy material where we have up to 5 micrometer sections available, and so we're saying that the experts were low for cellularity, so we would expect to get enough perhaps to start a DNA. However, some technology which may require more may have some difficulties. Okay, anything further, Jim?
No, I think you summed this up really well, thanks, Terry.
Okay, well then we'll move on to case 1. So with case 1, where we have a biopsy from a 71-year-old non-smoker. It's a bronchial biopsy showing the adenocarcinomas. And if we move up to the areas of the pieces, we can see better at this sort of magnification the individual pieces. If we call this piece 1, piece 2, 3, and 4, we can see that part 2 doesn't contain any tumour. However, the other 3 pieces do contain tumour, but it's well mixed within this inflammatory background and stroma content. So for annotation purposes, we would annotate, or the annotations would suggest annotating around each of the three pieces, either as three separate annotations or moving around with three pieces into one annotation. The tumour content that would result from that is difficult to assess, and for that reason the experts increased the range where we thought it's going to be somewhere between 11 and 30%. And I think this is typical of these types of biopsies where it is difficult to come down to a definitive category. So we know that it's probably sufficient in that it's greater than 10%, and for some downstream applications which may require 40% or greater, it's coming onto the borderline content. Jane?
I totally agree with everything, Perry. I would just say that it will be difficult to enrich to get it to the 40% because there's no clear areas of inflammatory cells or desmoplasia that you could remove, although possibly cutting ends off the endoscopic forceps pieces might be a possibility to get up to the 40%. But there's plenty of tumour spread around.
Yes, this sort of material could always be sacrificed on this fourth piece, but it's a bit more rich in this content, in this portion than the others. But when we get to cellularity, I think it's important that we increase the cellularity by including all of the other two pieces as well, even though they may not have as a rich tumour content. So for cellularity, we thought that the experts thought that there would be a medium amount of cellularity in this. Again, assuming 10 by 5 micrometer tissue sections. I can see where some people might think it's low. Certainly if we were taking less than 5 micrometer sections, it would be low. So certainly low to medium would be acceptable in the estimation of the cellularity. Jane?
Yeah, again, agree with everything. Obviously the tumour cells are quite large, so the surface area is not a good indication. So that's why we've gone with percentage and the medium cellularity, I think, when we were discussing it previously.
Yes, yeah. Okay, then on to case number 2. Case number 2 is a core biopsy of lung adenocarcinoma with lymph node metastasis. So this is actually probably a needle core from the lymph node. So we would immediately think of lymphoid tissue contaminating or diluting the sample. So if we go to a higher power and look, we can see very little lymph node structure in the tissue. However, the tumour is distributed well throughout. There are foci of lymphoid cells, which obviously are going to dilute the tumour DNA. So for annotation purposes, we would probably suggest that all of this could be submitted, even though there are areas. These areas we're looking at now tend to be more acellular, whereas at the end of the other piece, at the end of the tissue, there may be some inflammatory response. So taking it all or removing this end would be probably the way to go, and I think that's the way we suggested, and most participants agreed with that. This then gives a resulting cellularity, tumour content of between 1 and 20%. Again, difficult to assess, particularly when it comes down to if we include this sort of material where there's a higher concentration of lymphoid cells. as aggregates. So we are going for between 1 and 20%, taking the majority of the sample. Jane?
Sorry, Perry. Yes, I agree with everything you say, possibly chopping off the ends with the lymphocytic infiltrate, the lymph node that's remaining would help to keep the cellularity up, and the tumour content would be more medium than low then.
Yeah, and again, the resulting cellularity would be medium, similar to the previous case where we have large cells scattered throughout, no matter where we're putting that. So far again for the 10 by 5 micrometer sections. So Jane, anything further comment on this case?
No, I think that covers everything. Thanks.
Okay. Jane, would you like to guide me through the next two cases?
Right. So case number 3 for lung. Sorry, Terry, I'm on a really small screen, so can you read out the case details?
I will. I'm just putting it, I'm just trying to get the case. Oh, it's crashed. I'm going to have to restart. Can we pause it there, Heather?
This is case number 3 on the lung scheme, and it's a 74-year-old smoker, needle core biopsy with an adenocarcinoma. As you can see from the image, approximately 2/3 of this is tumour, with a third probably being normal lung. So the annotation shows the left 2/3 of the biopsy in the main, and most people agreed with all that. Then on higher power, as we look along the core, we can see that the nest of lung cells, the adenocarcinoma cells, are dotted along the entire length of the core. And if we could go up a little bit, that's good. There's a certain amount of desmoplasia, and also quite a few lymphocytes and lymphocytic infiltrate around the tumour. So the expert panel decided that it was approximately 1 to 20%, the same issues as last time, defining whether it was 1 to 10 or 10 to 20, because of the dispersion of the tumour cells throughout. There's also a little bit of necrosis which needs to be taken into account, but as it's in the middle of the core, that would be difficult to remove. Anything to add to that, Perry?
No, I was just going to comment on that necrotic area, because that is one of the things that a lot of people are concerned about, is including this necrosis. And as you say, it's in the middle, and it's very difficult to get outside of that. The only way, I guess, would be to sacrifice this portion. However, there are some good tumour areas within that portion. We could take it from there and take it from there. But I think this portion of tumour is probably worth the sacrifice of slight quality with the necrotic areas. But again, I think it's up to the participants with their downstream application how important they consider this to be.
Yeah, precisely. It depends on how much DNA you need for what you're doing with it, doesn't it, really?
Yes,
so,
and how, yeah.
So going on to the cellularity, the experts decided it was medium cellularity because there are a considerable number of nests of tumour cells throughout the core. And with the 5 by 10 or 10 by 5 sections, that gives you a medium cellularity and sufficient for most downstream applications. Harry?
Yeah, that's exactly what I was thinking, that it goes up to medium because of certainly with the numbers of cells. Thank you.
So I've got nothing else to add to case 3. Have you got anything, Terry?
No, I'm happy. Shall we move to case 4?
Let's move to case 4, yeah. Okay, this is case 4, which was supposed to be adenocarcinoma metastatic to a lymph node. And obviously, looking at this section, most people would realise that it's a piece of lung and not lymph node. So that's one point for people participating, is to ensure that the case history matches the tissue you have. Also, closer examination shows that there's no tumour in this piece of lung at all. So it's not just the wrong block from the correct cases, it's the wrong block altogether. Terry? Not a lot
exactly,
to say.
and I think it's a worthwhile point for these cases. Always read the clinical history, and I think clinical history in any case is the starting point. So clinical history, and does the description match the clinical history? And this is again one of the features that a lot of data sets within the college recommend, that the description matches the clinical history as an indicator of potential swapping of tissue. Otherwise, it's not for testing. Jane?
I'd agree totally, and also a call to anyone sending tissue elsewhere to be tested for good clinical history to follow the tissue. That's all for that one, thank you.
Okay, so I think that completes the lung study.
It does. Thank you very much indeed, Perry and Jane, and of course Heather from PathXL Phillips for driving this session for us. So if anyone has any queries, then please do get in touch with the UK MEQAS scheme, and you can contact us through any of the details on our website. And we hope you continue to participate in the lung cancer tissue eye EQA scheme. Thank you.
Thank you. Bye.